Hello, my dear friends. Today I am going to start the rest of the lesson, Chemical Basis of Life, the first lesson of the grade 10 science book, and I did up to here on my last video, and today I am going to do the rest of the part, start from proteins. Okay, it is easy to going our video with your textbook, and if you have your textbooks, please take it and follow it. 1.2 proteins. Okay, now when we are talking about proteins, it is the very important constituent in our body, and protein is an essential constituent of a human body and any other living cells. Okay, it has four major components. Okay, components means four major elements: hydrogen, carbon, oxygen, and nitrogen. Okay, this is the formula. This is the structure of one amino acid molecule. Amino acid is the basic unit of protein. Okay, basic unit is the amino acid. Okay, then this is the amino acid molecule, and it has four major elements: carbon, oxygen, hydrogen, and nitrogen. Sometimes it can include sulfur also, and this is the structure of amino acid molecule. Okay, then in here, it's we denote this one in here like R. Okay, we denote here as R. This R is equals to any hydrogen or carbon group. Okay, any hydrogen or it means R can be hydrogen atom, or any group of carbon. Okay, any group of carbon. R is equal to hydrogen or any carbon group. Okay, then in your book, the second paragraph, 17% of the mature human body, 17% of the mature human body is composed of proteins. Okay, that means 17% is the very big, the considerable percentage. Okay, when we consider our body, human body, 17% out of 100 is considerable percentage. Okay, then 17% of the mature human body is composed of proteins, and then they say protein is a complex molecule made up of polymerized amino acid molecules. Polymerized amino acid molecules mean please think if this one is the basic molecule. Okay, basic unit. This is the basic unit, and polymerized means the combination of these molecules like a chain. Okay, if you consider about some chain like this, this is polymerized unit. Okay, and we can consider just one unit as a basic unit, and this one is the basic unit of proteins, or in other words, of amino acid, basic unit of amino acid molecule. Okay, then this is the chain of amino acid, and we can call it protein or polymerized amino acid complex. Okay, polymerized amino acid complex is known as proteins. Okay, that's a simple definition for proteins: polymerized amino acid complex. Okay, then what is this one? This is the basic unit of amino acid molecule. Okay, then there is another thing. What happens when you have if H in here? If there is a H in here, H means hydrogen. Okay, if there is a H in here, then we can call it as glycine. Glycine. If H in here, you can call it glycine. Okay, glycine. Glycine means the smallest or simplest amino acid. Smallest or simplest amino acid is glycine. In here it has hydrogen, only hydrogen. Therefore this unit cannot contact with any other amino acid molecules. If there is a carbon group in here, then this one can link with any other amino acid molecules and can construct some complex chain. But with the H, with the hydrogen, it cannot link with any other hydrogen or any carbon groups or any other amino acid molecules. Then therefore this could be the simplest amino acid molecule. We call it glycine. Glycine. Okay. Then in here on the page number 8, the top of the page number 8, there is a small table for extra knowledge. Okay. Different proteins present in plants and animals. Okay. There is a very big variation in these proteins. Okay. With its formula and structure, but all those things are very important for the living beings, including plant cells. Okay. And in here, proteins present in muscles, proteins present in bones, proteins present in red blood cells, present in hairs. Okay. Lot of things in here. That's for you extra knowledge, and you should keep it in your mind. Okay. And let's come to the next part, significance of proteins. Okay. Significance of proteins. Okay. When we are talking about significance of proteins, it has very, very important significances of proteins for living cells, and living cells mean including plant cells. Okay. And what is the first one? What is the first one in here? Okay. First one, significance. The first one is as an energy source. Okay, we can use this protein as energy source, except carbohydrates and lipids. Okay, we can use these proteins as energy source, except lipids and carbohydrates. Okay, and what is the second one? To make structural components. This protein structure can be very helpful to construct cell membrane or any other cell structure. Okay? Then cell membrane is a very good example for that one. Okay? It is very helpful to making some structural components of cells. And the third one is as enzymes. As enzymes means, what is enzyme? Sometimes you heard this word before, enzyme. Enzyme is the thing that can speed up or that can increase the rate of biochemical reaction in our body. Okay? Enzyme is a thing that can which can increase the rate of biochemical reaction rate in our body. Okay, and as enzymes, all the biochemical check your book, all the biochemical reactions take place in organisms are catalyzed by enzymes. Okay? Catalyzed by enzymes. Catalyzed means I'll write in as a summary in here. What is first one? As energy source. Okay. And what is the second one? To make structural component. Okay. And the third one, as enzymes. As enzymes. Okay. Then when we are on the third one, as enzymes, you got new word in here, catalyzed. Okay? All the biochemical reactions take place in organisms are catalyzed by enzymes. Catalyzed means increasing the rate of reaction. Okay? Catalyzing means increasing the rate of reaction. Rate of reaction means the speed of the reaction. Okay. Then as hormones. Fourth one is as hormones. Okay. As hormones. Hormones are, I'm sure you heard that word before. Hormones are the form of proteins. Hormones are the form of proteins also. Okay. And the last one is antibiotics. Antibodies. Okay. Last one is as antibodies. Okay. As antibodies. What are the antibodies? Just check your book. The antibodies that are produced in the body to protect the body against microorganisms. Okay. Microorganisms that enter into the body are proteins. Okay. Antibodies are the purpose of the antibodies is protect your body from any other external microorganism. Okay. It can be bacterial infection or some kind of any other viral attack. And antibody means the antibodies are produced to protect your body from those microorganisms. Okay. Okay. Then we can come to the next part. Test. There is a little test to identify this protein chains. Okay. There is a little lab test, very simple one, to identify the protein chain, and we can see observe some color change from that activity. Okay, from that test. Okay, then this is the first lab test for identifying proteins. Okay, and we call it Burette test. Okay, we call it Burette test, and in here we have to take some dal solution. You can grind some little amount of dal with water, and you can make some dal solution, or you can use some egg yolk for this one, and you have to put that your solution, dal solution or egg yolk into test tube, and at first you have to put some drops of sodium hydroxide. Okay, then next you have to put some drops of copper sulfate into test tube. Okay, then you can observe some color change in this solution. Okay, consider if this one is a egg yolk. If this one is egg yolk, your solution turn into pinkish purple or dark purple. Okay, your solution turns into pinkish purple or dark purple. You can clearly observe that color change by doing this experiment at lab. And please memorize this one. This is a very simple test. You have to take a dal solution or egg yolk and put into test tube, and at first you have to use some little drops of sodium hydroxide, and then you have to use few drops of copper sulfate, and then you can observe the color change. Surely this egg yolk and dal solution are the protein rich component, protein rich foods. Therefore we can surely you can observe this color change by using this dal solution or egg yolk. Okay, you can observe pinkish purple or dark purple. Your solution will turn to this color, pinkish purple or dark purple. And then there is a little bit Okay, then there is a little part for enzymes again. Okay, for enzymes, on your page 9, the special proteins that are produced within the organism. Okay, it produced within our body. Okay, we do not we will not take that one from outside. Okay, it's within the organism to increase the rate of biochemical reactions in our body or in living cells. Okay, and it's known as enzymes, as I described before. And for example, there is a very good example for that one. For example, to convert sucrose into glucose. Now, you know these things: sucrose, glucose, maltose, fructose. Those things I discussed those things on my first video. If you forget that one, then please check my first video again. And for example, to convert sucrose into glucose. Okay, to convert sucrose into glucose, sucrose has to be heated with a dilute acid in lab. Okay, in our lab we have to heat that one with some diluted acid. Okay, to convert sucrose into glucose. But the enzymes present in the digestive system, okay, enzymes present in the digestive system in our body do the same reaction at low temperatures. Okay? Do the same reaction by low temperatures. That means it do not need to heat or do not need any extra heat for that reaction. Why? There is an enzymes in our body. There is an enzyme in our body that can, okay, that can increase the rate of that biochemical reaction, and then it can convert sucrose into glucose quickly. Okay, quickly. And there is another activity for activity of amylase on starch. Right? Activity of amylase on starch. Amylase also a form of protein. Okay, amylase also a form of protein. Okay, then this is also a very important, simple and important another lab test. Activity of amylase on starch. Okay, let's check that out. Okay, now in here we are test for amylase on starch in here, and amylase is an enzyme, and enzymes are belongs to the category of protein. Okay, enzymes are proteins, and amylase is a what? Amylase is an enzyme. Okay, therefore if there is a amylase, that means there is a protein. Okay, there is a amylase, that means there is a protein. Okay, now in here by this lab test we can identify amylase, the action of amylase, reaction of amylase on starch. Okay, starch is some extract of some dal or some any other cereal things, and it's normally it is in the powder form, and you can make some starch solution. Okay, you can make some starch solution by using water, and you have to get 2 milliliters of starch solution into test tube as a first step. Okay, as a first step you have to get the starch solution. Okay, the amount is 2 milliliters. Okay, you have to get the starch solution. The amount is 2 milliliters, and as a second step you have to add 2 milliliters of amylase into that test tube. Okay, that is the second step. 2 milliliters of amylase into that test tube, and then you have to mix it well. Okay, you have to mix it well. Okay, this is the handle, and you can mix it like this. Okay, and then after 2 minutes, okay, after 2 minutes you have to get some drop by using that certain equipment. By using that equipment you have to get some drop of this mixture. You have to get some drop of this mixture and put it onto white porcelain. Okay, why we are using white porcelain? We can observe color change clearly. That's the reason. Okay, therefore we can use white porcelain, mean white tile, piece of tile. It's okay. Okay, then you have to put some 1 drop of this solution onto the white porcelain, and then again you have to add some drop of iodine onto this drop. Okay, onto this drop after 2 minutes. We have to do this one after 2 minutes. Okay, then the color is blue. Okay, you add iodine onto this drop and the color is blue. Okay, then for a 20 minutes you have to do this thing, this the same procedure for a 20 minutes with 2-minute intervals. Okay, it means about 10 samples. You have to take out 10 samples from here and put it on a white porcelain, and then again add 1 drop of iodine onto that drop. Okay, 1 drop of iodine onto the drop of solution. Okay, then you have to observe the color change. Okay, now what happened at the first part means on a first drop and second drop and third drop, the blue color, at first it has very dark blue color, but with the time blue color reduces. Okay, it decreases the darkness. Okay, decrease the darkness, reducing blue color, and after that finally this drop, when we add iodine, this drop still remain in yellow or brown color. Okay, this drop still remain in yellow or brown color. What is that yellow or brown color? It is the color of iodine. It is the color of iodine. Then what can you say about that thing? Then we can say at the end of this experiment, that means that the last samples. Okay, at the last samples there is only iodine in here. That is the thing for this color. Okay, because this color belongs to this iodine. Okay, then what happened to the other one in this solution? What happens to the other one? It converts maltose. Okay, we take the sample from here. The sample means stark. Okay, in here it is a stark solution, and stark solution with the time. Okay, with the time stark solution converts to maltose with the help of amylase. We add amylase in here. Okay, we add amylase in here. Then with the time stark converts to maltose with help of amylase. Okay, amylase is an enzyme. Amylase is an enzyme. Enzymes belongs to proteins. Okay, therefore we can identify by doing this experiment, enzymes belongs to protein category, and in here enzyme we can identify and we can experience with the action of enzymes by using and by doing this experiment. Okay, and then in this part, okay, on this part you have to pay your special attention to the significance. This at the first you have to memorize the structure. Okay, structure of the protein, and you have to pay your special attention to the significances of proteins and what are enzymes. Okay, what are enzymes and what are the tests that you can carry out identify proteins. Okay, and then there is another part in this lesson, that is 1.3. This is 1.2, that one is 1.3, lipids. Okay, then we can meet on my next video with that part. Okay? Thank you.